Bonjour à tous et à tout le monde. Et je voudrais tout d'abord remercier chaleureusement Monsieur le directeur pour m'avoir donné l'occasion de présenter, de donner cette petite conférence. Et bien sûr, mon ami David Couvin pour l'organisation de cette visite à l'Institut, mon séjour à cette belle île, Guadeloupe. Merci, David. Et maintenant, je vais passer à ma présentation. Elle sera en anglais. Malheureusement, ce sera un peu plus facile pour moi, plus efficace. Alors, I will tell you about a very peculiar organism, which is named Prototheca. And at the beginning, I'd like to say that very few groups, very few scientists deal with this pathogen or even know about the existence of this organism. So I will boast about that. Our department, our group, is the only one in Poland and one of the 3 in Europe that are working on Prototheca algae. But I will explain the taxonomic position later on. But let me start with the general thing that most of us normally consider algae as very beneficial to the environment, as they photosynthesize and produce oxygen, etc. But again, quite a few people maybe know that they might be harmful and truly pathogenic even to humans and animals. And before I go to Prototheca, I would like to bring your attention to the problem of red tides, which are also known as harmful algal blooms, and which are caused by unicellular microorganisms known as dinoflagellates. This is a monophyletic group of algae that under specific conditions may produce toxins, and they are harmful really in the situation of massive blooms, so massive accumulation of the algae, and this goes with coloration of water, like here on this photograph. It's a red tide near Florida. And why they are harmful, these algae and these toxins. So as I said, they produce a number of toxins, of which these are the most dangerous. So these are neurotoxins. And as you see, different toxins are caused by different species. And I would like to right now show you the producers of these toxins. So Alexandrium, Karenia brevis, Pseudonychia, it's in fact diatom, it's not strictly dinoflagellate. And these intoxications are after exposure to these toxins. Humans suffer from different syndromes after eating different types of shellfish, seafood, so mostly clams, oysters, mussels, etc. And the symptoms occur something like several hours up to 2 or 3 days. And most dangerous symptoms are paralytic shellfish poisoning, and amnesic shellfish poisoning, as the names suggest. These toxins, saxitoxin and domoic acid respectively, can produce paralysis and amnesia. And with these symptoms, several death cases have been recorded. I will not dwell on the chemistry of these toxins and even organs, because it's not the main point of this presentation. But I want just to say that the harmful effect is through toxicity of dinoflagellates diatoms. Also allergenic can be some algae that are found in dust, for instance. So species of subgenera like as Ankystrodesmus, chlorella, chlorococcum may cause skin allergic reactions, and these species have been linked with the symptoms of allergies. However, while with these toxins or allergies caused by the algae, it's not that we can say as pathogens. Very, very few algae can be truly pathogenic, meaning they can enter into the body and cause a disease, infection. And here I would like to give you words on three genera that of the algae, very unusual organisms, which might be pathogenic to certain organisms. So the first one is, and the first had been described in 1921, was Helicosporidium. It's a colorless alga, and it's unicellular, of course. It's a pathogen of insects, mainly larvae of insects of the orders Lepidoptera and Diptera. And this is obligatory pathogen, but infections can occur only in invertebrates, yes. Then comes Comixa. It's also a pathogen of invertebrates, but specifically mussels. So, and contrast to Helicosporidium, Comixa is photosynthesizing, and you can see in this section of the gland section some details in the sequence with this algal foci, green foci. This is just an infection. Then Chlorella, discovered in the 19th century, but first infections in animals were reported in the 1970s. And Chlorella, you know perhaps very well, this algae is very, it's omnipresent, it's very common in the environment, but rarely it can cause infections, and it is in animals. So far, only 2 infections in humans have been reported, with almost spontaneous recovery. So they were moderate, or not very dangerous. The people, but the cases in animals like cattle and sheep can be fatal. Also, a single dog, gazelle, and camel have been infected with Chlorella species. the mechanism through which chlorella, and this is also the case in Prototheca, I will talk about later on, are unknown. Why normally photosynthesizing alga starts to be pathogenic. It is thought that the animals get infected with chlorella after exposure to contaminated water sources, and the disease ranges from superficial cutaneous or lymph node infections to multi-organ dissemination. So it might be chlorellosis might be severe in animals, as I said. And finally, our Prototheca, which is much more frequently observed as chlorella, as a positive agent of infections in both humans and animals. So I'd like you to know that chlorella and Prototheca are the only algae that are pathogenic to vertebrates, humans and animals, while chlorella infections are very, very sporadic. In humans, 2 cases. Prototheca, much more common, and I will show you later on how many cases, because I'm dealing also with this, we have had so far due to these organisms. But first, some facts pertaining to Prototheca taxonomy. So I just showed some few breaking discoveries, let's say, because the matter of Prototheca taxonomy has been very controversial, and the taxonomic position has long been discussed. So first, 1880, it's the first isolation of these organisms by Zoff and Kuhn from slide flags of lime and elm, generally these 2 trees. They just noted these organisms, but did not describe them. This was done by Kruger in 1884, and he described 2, he called them fungus-like species, because they were colorless and they were very similar in macromorphology to fungi. And he called those 2 species Prototheca zophi and Prototheca morphoides. And so, according to Kruger, these were fungi. They were more closely related to fungi and to plants. In 1904, Behring, the famous microbiologist, observed a spontaneous loss of chlorophyll in Chlorella variegata. Thus, the idea of Prototheca being achloric mutants of Chlorella. Also, Chaudin, in the meantime, in 1913, pointed to the similar reproduction, Prototheca and Chlorella algae. But then some colorless strains of Chlorella, this time pyrenoidosa, were obtained through irradiation. So at that time, in the mid-20th century, the concept that prevailed was that they have nothing to do, in fact, with plants, with algae, because they are mutants of Chlorella. However, this standpoint was questioned by Ciferri in 1956, who compared the nutrition of Prototheca species and artificially produced Chlorella strains, those colorless strains, and he found that his nutrition patterns are completely different. For instance, Chlorella mutants were able to synthesize thiamine, while Prototheca were unable to do this. This is just one example. A crucial point in this discussion, in this history, was a comparison between Prototheca and Chlorella protothecoides, which was described nota bene by Kruger also at the end of 19th century. This was quite neglected species. So these comparisons, which were performed at the mainly physiological and biochemical level, showed many similarities between the two organisms, yes, Chlorella and Prototheca. These similarities appeared entirely in the lack of use of nitrate as a salt, nitrogen source, high salt tolerance, so lack of hydrogenase activity, and many others. So due to this closeness of Chlorella protothecoides and Prototheca, the former was reclassified as Auxenochlorella protothecoides, and this gave us spur to further research of the two algae, let's say, already, and these research reached the molecular level. Crucial were studies performed in the 1980s and early 1990s that focused on comparisons of RNA sequences, and eventually these studies showed unequivocally that Prototheca species are much more related to plants than to fungi, and that these Auxenochlorella protothecoides might represent first closest ancestor to Prototheca gens. And also I would like to mention about observations of Melaka-Bukharin and McCracken, who used electron microscopy to demonstrate presence of certain structures that resembled chloroplasts. They were eventually described as vestigial plastids, and we do find plastid DNA in Prototheca. Another argument for the taxonomic position as plants, in fact. So here's how current Prototheca taxonomy looks like. So there is now no discussion over this taxonomic position. So they are placed in Viridiplante, Chlorophyta, class Streptococcus, and Chlorellales order. And we know 18 species. Those marked in red are those implicated in disease, humans and animals. The others are of environmental origin. Here I would like to show you a phylogram which shows this position of Prototheca among those other algae like Auxenochlorella, but also Helicosporidium, which is also considered an alga and plant. This phylogram was inferred from partial cytochrome B gene sequences, and what we see, what we see is that the non-pathogenic species, those environmental, 3 here only, but the other is in the same case, form sister lineages, separate from other Prototheca species. We see that Prototheca strains of Prototheca bovis, Chiferi, and Blaszke, those pathogens, mostly of the cows. I will talk a bit later on about this, are very closely related and share monophyly. Then that the Prototheca vicarami is more closely related to Auxenochlorella than to other Prototheca species. So it is postulated that Prototheca form separate genus even. And the whole lineage has been described up in this phylogeny, which includes Xenochlorella, Helicosporidium, Chlorella, and Prototheca. And now something about biology of these algae. So first, the habitat, so where we can find these algae. So frankly, they are thought to be everywhere, and most publications repeat this view that Prototheca are ubiquitous. We have slightly challenged this view. So where we surely find them is slight flux of herbaceous plants and some woody trees, but mostly in water. Freshwater courses, lakes, artificial water bodies, and ponds, swimming pools, fish ponds, and what's quite interesting, I think, they can be recovered from domestic and municipal sewage. And very interesting the fact that they resist different disinfection procedures like chlorination, so they can simply enter to the environment from the sewage treatment plants. Soil, but here we somewhat question this omnipresence in soil, because performed a study very recently which showed, in fact, at least in Poland, soils, a lack of Prototheca in the soils we have isolated, and it was mostly mud, and soils with a high organic content. A few strains out of something like 500 samples. Human and animal feces, Prototheca are known to pass through the alimentary tract without causing any harm, and excreted with the feces. And food products, beef, pork, also seafood and dairy milk. As you will see in a moment, Prototheca are very closely associated with dairy cows and dairy industry. Metabolism. So these algae are aerobic, heterotrophic. I forget to tell you that, for instance, dinoflagellates are mixotrophic. It's interesting. So they are, many of them are photosynthesizing, but they can also just ingest prey, and this is called phagotrophy. But Prototheca are heterotrophic, meaning saprotrophic, and sometimes pathogenic. A wide range of carbon sources are utilized, so alcohol, carbohydrates, assimilation occurs, and there's a lot to do with diagnostics. You will see later. Glycogen as a storage carbohydrate. In Chlorella species, this is starch, which is neither present nor assimilated, or not can be assimilated by Prototheca. Ammonium chloride, not nitrate, which I mentioned, and some organic compounds and acids as a nitrogen source, and this dependence on exogenous thiamine, which was same was for oxynochlorella, as you remember. And very nice is the morphology of this algae. They are, as I said, I hope so, unicellular algae, and they are these cells are spherical to spheroidal, size 3 to 30 micrometers in diameter, depending on the species and developmental stage, and they reproduce asexually through a process called multiple fission. So it's just the same mode of reproduction as chlorella species. It's called also endosporulation because just when we have a single cell, it undergoes several divisions, and everything occurs within the maternal cell wall, and we have and then we have such structure like sporangium. So here you can see the sporangium with 3 endospores, so sister daughter cells, and they are released upon rupture of the cell of the mother cell. So these are really nice pictures. I'm very proud of them. Here you can see a cross-section of the Prototheca cells, and this transmission microscopy, and you see 1, 2, 3, 4, 5, 6, 7, and 1 centrally positioned daughter cells within the sporangium. So it's it's called also a daisy-like structure, especially in Prototheca bicorami. So it's quite related with species. And these structures, again, in the scanning microscopy, the former photographs, this is very similar, I think, to morullas, and this structure is called morula-like. Another aspect of Prototheca morphology, in fact, is the cell wall composition. So it, of course, does not possess cellulose, as chlorella, but different sugars whose key constituents are glucose, galactose, and glucosamine. Also, they contain sporopollenin. It's a very rigid polymer, also seen in pollens and spores, fungal spores, for instance. And it is believed to account for up to 20% of the cell wall. So, and sporopollen is thought to be responsible for this rigidness of the cell wall and resistance to different chemical and physical treatments. Still, it's not, of course, cell wall constituent, but it's present in the cell membrane. And it's quite interesting because this the aerosol we have in the fungal cell membrane. And this will have consequences with treatment options, for the treatment options. Now there is pathogenesis. Why and how are Prototheca algae pathogenic? So this matter is largely unknown of the research concerning on the pathogenesis, and let's say in statu nascendi. So they are really in the infancy. So what we know is that Prototheca can easily colonize skin, nails, and respiratory alimentary tract of humans and animals. That the infection occurs mainly through contact with contaminated sources, and often some trauma occurs. So infection is through traumatic implantation, microbiology from the environment, but exact portals of entry are not defined, and we are still trying to look at them closely. What we also know that no animal to human nor human to human transmission has been documented. That the incubation period is from weeks to months. That the lesions generally remain localized, but they may spread hematogeneously or through the lymphatic system to other organs. And this occurs, so the systemic form of prothecosis usually can be observed in severely immunocompromised patients. Finally, immunological aspects of Protheca infection are poorly known. What we know is that the immunological response involves chiefly neutrophils, macrophages, and natural killer cells. And we also know that some defects in neutrophil activity is crucial for the host's response to Protheca infection. Defects in neutrophil functions may facilitate the infection to occur. Low virulence in animal experiments, so it was very hard to infect, for instance, neutropenic guinea pigs or athymic mice experimentally with even high doses of Protheca cultures. And this is in favor of the view that Protheca are generally of low pathogenic capacity. And I would like to show you some clinical presentations of the disease. So prothecosis exists generally in three forms: cutaneous, articular, under the form specifically of olecranon and bursitis, systemic, and here are some here are some pictures which show how these infections may look like. So here we see edema and bilateral lack of epidermis. These are some erythematous lesions on forearms or arms. These are pustules and fistulas on hand. Also, Protheca infection can manifest as infections of fingernails. So we see discoloration and hyperkeratosis of nails. This is pyodermic infection of legs, and some lesions also on legs too, and here on face, cheeks, jaw. So these are mainly cutaneous manifestations. And here I would like to show you with our quite recent analysis, which shows how many cases and due to which Protheca species we have. Cases of prothecosis, of course. So they were mostly reported from USA, also from the Asia. In Europe, they are quite rare. In Poland, we've had only one case. And as you see, most of the cases are due to Protheca vicariatus, the key human pathogen among Protheca species. Overall, there have been 252 cases, and mostly cutaneous manifestation, 15% disseminated form, and the others just smaller proportions. But what we can see now is an apparent increase in the occurrence of Protheca infection. This is due to our advancements in the diagnostic schemes, and also greater awareness of this clinical the disease itself. So while at over 30 75 case reports in last 2017, but the 20th, the number doubled almost. Treatment. So there are, in fact, no treatment options for prothecosis. It may some very, very mild cases, problems can just subside spontaneously, and we do not need to do anything particular. They are just to use supportive treatment, but nothing special, specifically targeted Prototheca. But in some more severe cases, antifungals are used, and this is because of that ergosterol I mentioned as a strength of cell membrane. Since most big part of antifungals, like the azoles or amphotericin B, target the synthetic pathway of ergosterol. And these agents sometimes do work, sometimes they don't. It's quite complicated, and the results of drug susceptibility testing, in vitro testing, of course, may not be linked to clinical response, sadly. Then, so what are the treatment outcomes? Prothecosis. So among those 252 cases, half of them have been cured. And 13% there was improvement. So in large part, we have just successful treatment. But still, in something like 30%, it's quite high as for the opportunistic pathogen, we have treatment failure. And here you can see the outcome of some selected cases which involved different systems, different organs. So, of course, among patients with systemic disease, the death rate is the highest. And in such cases, it is often observed, of course, in people with severe immunosuppression, mostly. In such cases, we observe that no drugs are helpful. So just supportive treatment again, and waiting for the response. I do not want to shock you with this, I'm clinical microbiologist, so I couldn't resist, sorry. Just to say that in animals, and especially in dogs, the disease is very severe, and we do not know why. Some host-specific factors may be responsible, but we did not identify them. But anyway, scaling prothecosis is mostly systemic disease, and it develops very quickly, and it involves different organs and tracts, like gastrointestinal tract, genitourinary tract, nervous system. Blindness is common, paralysis is common, and here you can see the diagnosis. And the key agent of this case is also, as in humans, Prototheca vicarami. Again, I mentioned that generally in animals, Prototheca is thought to be transferred into humans through contact with some water sources contaminated with the algae, and often trauma makes the portal of entry for the pathogen. The disease has been also described, apart from dogs and cows, are the key animal hosts, also in cats, goats, fruit bats, beaver, deer, single cases, and horses. And a bit about protothecosis in animals, because it is a really great problem. I'm sure you've heard about mastitis, yes? So this is the most commonly observed disease of dairy cows. And the mastitis, as long as considered, caused only by bacteria and fungi, yeast mainly, and bacteria, Staphylococci and Streptococci, mostly. But since 1952, in fact, there's a range of pathogens brought in by Prototheca algae, and the increase of this bovine protothecosis is occurring. So the disease is on the rise worldwide, actually. Of course, in some countries which provide very detailed control of the herds, there are found much more cases than in those that with poor diagnostics, with poor overall poor microbiological control of the herds. So what are the sources? So this is the disease manifests as normal, let's say, mammary gland inflammation, so mastitis. It can be clinical or silent, so subclinical. And the key agent here is Prototheca bovis, at a lesser degree, also Prototheca blaszka. Sources of infections, again, different environments, different surrounding the cows. So mouth, manure, milking areas, etc. You've got this slide. The infection can be transmitted by during milking. It's quite often through the milking machines, or by milkers' hands, also from cow to cow, of course. The risk factors are age, so the older the cow, the greater chance for the development of the disease. Number of calvings, again, the more calvings, the greater chance of infection. Past inflammatory processes, different infections, and of course, poor milking hygiene, so lack of proper practices of disinfection of the teats before and post milking. No effective treatment options available, and bovine mastitis is normally drug resistant. So, or it can be cured with perhaps not cured, tried to be cured, but with by means of some routine agents, but there's no sense in the economic sense. Okay, so while the cow is sick, the milk production drastically drops down, and the cows are separated, but eventually they are calved. So it's very sad, but it's this. Yeah. So. And the milk of these cows, if still can be given by the cow, is of watery consistency. It's white to yellow with flakes, with pus, etc. So it's for nothing, in fact. Now, diagnosis. If I speak too long, please hurry me up because I'm it's okay. Hope you are not boring. So a diagnosis. Normally, prototheca can be diagnosed upon histopathology examination. We use standard staining procedures like with hematoxylin Elzyme, but even better works periodic acid sheath or staining with silver, and you see very pathogen, sorry, this word I can't. Anyway, very characteristic structures for Prototheca. These daisy structures, these sporangia, can be easily observed. Microbiology, it's also quite easy because Prototheca grow on variety of media. Blood agar or subrod agar is a very simple medium for fungi. On SDA, on the subrodextrous agar, growth occurs after up to 48 hours, usually at 25 to 37 degrees Celsius. Also, we have Prototheca isolation medium when we deal with some heavily contaminated samples, mainly environmental origin. Staining, I said, is biochemistry, so different species are identified through carbohydrate and alcohol assimilation profiling. Also, of course, it's not normally used. Electron microscopy can be in assist diagnostics. Also, of course, some organisms like yeast candida, Ucella is very similar. Macromorphology with Prototheca can be differentiated, for instance, by using chromagar or on Christensen's agar with urea. Candida species hydrolyzed urea are while Prototheca does not exhibit such urease activity. Of course, some dimorphic fungi in their yeast phase can imitate Prototheca, but to be sure, if we are dealing with Prototheca or dimorphic fungus, we can simply set dimorphic cultures. So we culture both 26 degrees Celsius, 37, we've got yeast form, while in 25 we've got mycelial form. And here, different, as I said, estimation of different carbon sources is useful to differentiate between Prototheca species like Prototheca piperami, bovis, chiferi. But now normally we use genotyping. And here are some key questions, some key issues related to Prototheca and protothecosis. So we study prevalence of the disease in humans and animals. We look for the pathogen reservoir, carriage, source of infection, also the modes of transmission, and we try to delineate risk factors. What is unknown, I've already mentioned a bit about it. We do not know how saprotroph becomes a parasite, what are the mechanisms that drive this conversion, and how the colonization turns into infection. So again, what are the mechanisms responsible for this? What are the virulence factors and what are determinants of drug resistance? And in our department, we deal with these three fields related to protothecosis. So diagnostics, treatment, and epidemiology. here are our aims of our studies. If you allow, I will just briefly show you what we how we do our research and what are the results. Just need some more minutes, okay? So while studying the epidemiology of Prototheca mastitis, we do survey among dairy farms in Poland. Based on history of mastitis, based on medical documentation, and of course we visit those farms, and first what we do is we examine milk with California mastitis test. This is wild cow test performed on such plates, and we simply take some milk sample and add a specific agent which makes clots with immunological cells, like neutrophils. So if the number of these cells indicating that inflammation is high, it gets dense, and we know that the inflammatory process is ongoing, and it might be Prototheca, but also some other infection with some other agents as well. And of course, yeah.
What is a BA on the data on the right?
BA, what is it?
Gram-negative staphylococcus.
Yes.
BA.
Here is a gram-negative BA. Oh, this is. I must think a little bit. Staphylococcus BA. That's a good question. I will just think a little bit. Okay, can we get back to this later on? Okay, just sorry. And we take also to pictures for the colonization, like swabs from the animals, also from the healthy ones, and from the cow's environment. So feed, manure bedding, soil surrounding the barns, take a lot of samples, and we perform farm survey. Just test different factors that may somehow related with the infectious process. So here are some results. So we do our research regionally. So this is one of the voivodeships, regions of Poland, which we tested. And as you see, we found Prototheca in 13% among environmental samples, and 8 sorry, 4 and 0.6 in among samples from the animals. The nationwide scale, we also did this study, and the results as you see. So overall, 8.3% of milk samples were Prototheca positive, and the other percentages given here like the prevalence of Prototheca among other samples, environmental samples, or swabs from different anatomical sites. So again, this is how the species, in fact, the etiological agent structure of the bovine mastitis look like. And here the Prototheca species structure among the Prototheca positive swabs and environmental samples. I will just try to summarize here these findings. So the overall prevalence is something like 5% and 8% at regional or nationwide level. So it's not that high. Prototheca species were third after Streptococcus and Staphylococci, most common mastitis pathogens, and most cases were subclinical with a high somatic cell count, but insignificant blood cell counts. Prototheca bovis was the key pathogen, and also Prototheca bovis predominated among samples from different anatomical sites, and also among environmental samples. However, among the latter, also other species like Prototheca chiferi and Prototheca blaszke. As you know, Prototheca blaszke may also cause the mastitis. Did occur in those environmental samples, and anatomical size of the cows, yet at less frequency. Yes, and no other Prototheca species could be observed, so this suggests that they have predilection for some different ecological niches, perhaps. Yes, and so important. And the second of the three areas of our interest is searching for new Prototheca agents. So we perform a lot of susceptibility testing using both old agents or even neglected agents, but also some newer ones. Here's just a summary of what we know for quite a long time about Prototheca resistance. So they resist certain enzymes, tolerate a wide range of pH, and salt, up to 18% in Prototheca blaszke. And what we observed is also resistance to high temperatures, including pasteurization treatment. And poor susceptibility we normally see when we use antifungal agents, even those newly introduced to the market. So here are some results. I will not dwell on this, but still the most active is amphotericin B, but as you perhaps know, this drug is causes nephro and hepatotoxicity, so it's to be very careful while using amphotericin B, but it's still a drug of choice in the treatment of, for instance, systemic infections in humans. Here are some experiments with the results of some experiments with high temperatures and disinfectants. We showed, for example, that iodine, which is normally used as a disinfectant, can be used at much lower concentrations. We also tested three new substances, say new in the field of protothecosis substances. So one of the group of ionophores, 3-bromopyruvate, which is a promising agent in patients with cancers, and is often very little toxicity. And silver nanoparticles were also tested to see if they can be efficacious against Prototheca species. We used different cell lines to test those agents, like mouse fibroblasts and bovine mammary epithelial cells. We also do the checkerboard assays to analyze the possible drug interactions, etc. So here are some perhaps dull results for you, so I will just these are cytotoxicity tests. I will just summarize everything in one slide. We also study the activity of nanoparticles at the level of microstructure of Prototheca cells. So, for instance, we showed that upon action of silver nanoparticles, the cytoplasm shrinked, and the effect was lethal to the Prototheca cells. Oh, sorry. And these are the key findings from those slides. They are susceptible. Sorry, it should be resistant, in fact, to most of the conventional antifungals, but the highest activity, if the drugs are active, highest activity is observed for amphotericin B. Those three substances I mentioned are promising because they showed anti-Prototheca activity, and particularly silver nanoparticles had the highest algicidal effect, given that the MIC and MIC were equal. Prototheca bovis are the least susceptible, so the most resistant, and this may relate somehow, with the pathogenic potential of this algae, at least towards the animals like cows. Iodophores were shown promising and was hypothesized to replace standard iodine solutions as less toxic. Though all three substances, iodophores, 3-bromopyruvate, and silver nanoparticles, may be potentially used as intranasal preparations for legitimate Prototheca mastitis. It's very difficult, as you may imagine, to test in vivo those new agents, as we have very, very strict procedures for the good of the animals. So it's many commissions, and it's really cautionary to go with it, but we shall try thinking of it. Under different formulations, these substances may be used as treatment of at least superficial infections of animals and humans. And 3VP, since it showed previously success while treating oncological patients, and it was shown as having very low cytotoxicity, and it is the most promising agent for the systemic treatment of prothecosis. So it's still to be discovered, the effect. And finally, a few words on diagnostics. So we started genotyping. These genotypes 1 and 2 of Prototheca zopfi are currently Prototheca bovis and chiferi respectively, so it's completely changed. It's just a slide from our very early experiments. So it was first biotyping, then genotyping, based upon 18S rDNA gene partial sequence. But now the standards, these are first results with Uwe Rössler from Germany, the results of identification of Prototheca species. We also tried Malditov mass spectrometry to identify different Prototheca species, and we managed to set the reference spectra for 8 species, including 3 most commonly observed, at least on dairy farms. There are some kilograms showing the relatedness of Prototheca species, allowing differential diagnosis based on large and small ribosomal subunit sequences, gene sequences. But what we now use, and what is let's say, gold standard, which I'm very proud of, this new marker, side B, a relatively new marker since 2018, which allows delineation of all Prototheca species that currently exist. So this kilogram, you know, from the beginning of the presentation. And we, based on this marker, we also, to avoid costs related with sequencing of this gene, we devised a new method, simple one, PCR-FLP, that assists identification, and it nicely shows, identifies all the species, okay? We also focused on, well, in fact, we entered somehow the genomic level as one of the first, yes, the first, I think, of those Prototheca investigating groups. So first we invented thickest isolation method of DNA. Because, as I said, the Prototheca cell was very rigid, and it was very hard work really to get inside of the Prototheca cells. So after many months of combining different agents, different isolation strategies, we did it, and we got a method that is that nicely works for obtaining total DNA, also with the aim of sequencing Prototheca genomes. Yes. And last thing regarding diagnostics is that we consult and are involved in diagnosing different cases of Prototheca disease, both in animals and in humans. So we were among the authors of this case report, the first, the only one, Protothecosis in human. It was a child with hydrocephalus, and in fact, it's said that the Prototheca infection, it was a neuroinfection. The Prototheca was isolated from the cerebrospinal fluid, and the Protothecosis was in fact cured, but the child died of this hydrocephalus. And also we described the first case, it's confirmed case of infection of a fish, in a carp. It was due to Prototheca vicerami, as that case in a human. And all this whole genome sequencing, so we what we did and still do is want to explore genomics of protothecology, and we so far done only sequencing of the Prototheca vicerami genome. It was a very preliminary study, and still these are our targets to get a detailed picture of the genetic makeup of the species, not only different Prototheca species, and find out molecular determinants of pathogenicity and drug resistance, or some new molecule, perhaps even better than site B, phylogenetic markers for identification or evolutionary studies. And this all is directed at development of new diagnostic assays and therapeutic approaches. So these are just very few selected observations from the very, as I said, preliminary analysis of the Prototheca vicarami genome. What's very interesting is that about 40 genes that are unique for Prototheca vicarami, and we found some genes that share similarity with those responsible for pathogenicity in fungi. Yeah, in summary of this part, I think I said everything, so perhaps I won't. I will skip it. But I'm just 2 slides more. Sorry. Reaching the end. So on this slide, I would like to say what is still underway in our laboratory. What we find, so it's continue these environmental studies, epidemiological studies, and what we also do is very nice research project on infection models in mice and in eukaryotic cell lines. So we want to perform adherence assays, infection assays, and observe macroscopically the effects of experimental infection in mice, immunosuppressed, nude mice, and normal ones. So it's in progress, and we expect the results in late this year. And those genomic studies, and this is what I would like to interest of yours. Since we truly lack specialists, experts in genomics, who could help us with assembling genome sequences, perform functional annotations and gene predictions, or make some phylogenetic, more advanced phylogenetic analysis. Still need to determine precisely the gene protein functions, also the proteomics. It's far ahead of us. It's also here do not have experts in our lab, in our institute even. So if you can somehow help this meeting, is also to perhaps get into closer contact, into sort of collaboration. I'm very, very open and I would be very grateful if you could think of it. And the last slide is greetings from Poland. Yes, so this is our faculty of biology. This nice picture showing the old building of the university, which is completely in other part of the city. David was there. And it's the address, we've got website of our department, and here's Zofia, who sadly, as David said, could not be with us due to some health problems. Professor and servants, 3 PhD students, and his profile and information are going to just make you willing to come here. It's not so cold. And yeah, that's it. So merci pour votre attention. Sorry for this long speech. Sorry. Thank you. Some questions, perhaps.
Yes. Si vous avez des questions.
What's the method used for finding the pathogens in the samples from the globe?
From where? From cows. So the same methods as in the case of human infection. So we use histopathology. So we take samples of the udder, but normally it's of course testing the milk first of all. So microbiology studies, microbiology and histopathology.
Culture.
Yeah, culture. Yeah.
Yeah,
I'm thinking of it. I'm thinking of it.
No?
Sorry.
Bacillus anthracis.
No.
No?
No. bacillus are very, very, I think they are not at all etiological agents of mastitis. So this has to be, I will let you know just as I learn. No.
Did you have the possibility to test other animals from Europe, not only in Poland?
Yes, of course, and this is also what I would like you to encourage to collaborate with me, because we are very interested and we collaborate with different countries to assess the prevalence of mastitis in dairy cows. For instance, very recently, I've been contacted by a guy from Bangladesh. So we will perhaps start the study on samples from there, and also from the UK. So we are in touch, and I don't know how the dairy industry functions in Guadeloupe, for instance, and how it is developed by. We can also, I will be very happy to test some samples from here, from the Caribbean islands.
Not in the US, because we observe a lot of people contaminated in the US. You don't have samples from US?
No. They have, in fact, their own groups, and in the US, the awareness of the problem is rather high, because the problem is not only with but a large proportion of Prototheca cases in humans is from the US, but also in many dairy herds the problem is known, it's dealt with, and well, I do not look for collaboration because they are just from
For your genomic studies, you use only NGS or also long reads?
Long reads also. For instance, the Prototheca vicerami species was analyzed through Illumina and PacBio. So PacBio, I think, is long reads. Yeah. Yeah.
When in your study in pandemic, in one sample, when you find a Prototheca, did you find a bacteria in the same time?
Yes, it can happen. It's not very often, but this co-infection, protothecal bacterial, may occur.
But sometimes there is just Prototheca, or
yes, of course. Mostly, if we find Prototheca, we find it solely as a sole etiological agent, so sole pathogen in the milk samples in the udder. Okay. Of course, if we have co-infection, we cannot be sure as for whether the clinical signs are due to more to bacteria or to Prototheca. I think in such co-infection cases, the cases are rather clinical. So you see the swelling of the udder. It's a really large one, and the inflammatory process is very intense. While on the Prototheca, it's usually subclinical. So it's longer, more persistent. It's still resistant to and the effect is the same. The lack, the restriction, or the prevention of milk productions.
Is your protocol for DNA extraction with Prototheca published?
Yes, we got this paper.
It works on fungi or so?
I think so, because it's it's in fact a combination of different protocols used for fungi or other algae. So it's a mix of different isolation schemes. So I think it would be it might be even too harsh. No, perhaps not for fungi, because the cell is also quite rigid, but it should work. Yeah. It's still not a snap. We have this isolation procedure, but still, it's you know, some challenge to isolate it. We obtain it, but you have to be very careful, and you know, it takes some time. Yeah, but it works, yes. And as for Prototheca vicarami, we could just take advantage of this isolation method to those genomic studies.
When you found Prototheca in milks,
Mhm.
do you have to test again the farm, or it's okay, people can drink the milk?
So that's a good question, because it's still a controversy, because you know no disease has been observed upon drinking milk drink. Yeah, but still it poses a potential hazard. Okay, so it's much better to eliminate Prototheca from the milk, but as you know, sometimes it can resist those pasteurization procedures and heat treatments. So and we could find some Prototheca after this treatment. So well, for instance, yeah, yeah, nothing.
Okay.
And the taste is the same?
Yes, the taste of this milk, which might contain Prototheca, is the same, because if they are found at very low concentrations. But if we, of course, take milk from cow with those signs, or even subclinical cow, you saw how the milk looked like. So it was, of course, unable to be put on the market. So, but sometimes normal milk in the shop can contain Prototheca, and no, as I said, no case has been found of any not disease, but any clinical changes or disturbances after drinking milk. So hopefully it's harmful, but generally the idea is to prepare milk to the market free of any pathogens. Yeah,
They can contain small concentration.
The regulations. We do not have regulations concerning the Prototheca content in milk, because those studies are quite random and very few. So I can nothing about it. I think as clinical should be no Prototheca in the milk. Okay, if it is, I wouldn't drink it simply. Okay, but we do not have any regulations on that.
So contamination has been proved by culture, or also metagenomic analysis?
No, we did not do any metagenomic analysis. So those contamination was evidenced only upon culture.
And 18S may work on
It may work, but only for certain species. If you mean identification. Yeah. Yes. So even more, I would say that while the current standard is that site B gene, to differentiate between two quite important species, because Prototheca bovis and Prototheca schiferi, you have to sequence this site B gene to be sure. While for these two particular species, sequencing also of the 18S is sufficient, so fine, okay, but not for the other, for instance, Prototheca vicarami. So I think the new marker could be useful, but that the one we have is also fine. Just to be completely sure, we normally, if we bank species, deposit the sequences, we do the sequencing simply. It's the yeah. Sometimes it's
because we have
okay.
realized the study on the source of Guadeloupe using 18S.
18S, okay.
Genomic, and maybe we could find. We didn't check for Prototheca.
Okay.
We were looking for Lima. So
I think it can be detected somehow, but well, with different, no, I'm sure absolutely different primers than for bacteria or some parasites. I would say it wouldn't work for Prototheca.
So you were interested also in genomics. I will not discuss the genomics. Maybe after lunch, if
Okay.
you're available,
Yeah.
I can show you what we do.
Yeah.
Like I told you earlier, we are not an expert yet.
Okay.
We are willing to become,
Yeah.
but at least I can show you what we've done so far.
Great. Yeah.
Okay.
Yeah.
So
Great.
because we struggled a lot at the beginning, now the pipeline is kind of set up, and we are willing to go further on the analysis.
But I think that your aims are quite similar with mine, yeah? With different pathogen, but still the methodology can be.
Periodic, so
That's yeah, exactly.
But I will show you afterwards.
Thank you
because
very
I'm not
much.
going to go into details.
Yeah, yeah,
Okay.
great. That would be awesome. But also think of some guys who could just perhaps collaborate with me in this epidemiological study, because it would be really nice to know. I don't mean only cows, but also some environmental sites. I would say that Prototheca, there's a greater chance to find Prototheca in sea around the islands here than in Poland, because it prefers some warmer, in fact, climate zones.
You were talking also about water, right?
Sorry?
You were talking about the presence of this algae also obviously in water. Algae, water. So we don't have the results yet. But we did a lot of sampling on waters, mostly rivers. So maybe after the metagenomic analysis, we are doing 18 tests. But the problem is we are cultivating first. So because to have our analysis done, we first have to cultivate the amoeba. So we don't have the original sample. I don't know if the culture conditions that we use are suitable to detect your algae. So maybe it won't work for the soil.
But sorry, why don't you have the original sample? What do you mean?
Because it's not enriched enough.
Okay.
We have to cultivate it first. For the amoeba.
Okay.
We tested it in soil.
But do you think you would have a bit of this sample to simply culture for Prototheca or not? Oh, no. That's possible. Okay.
We have the originals.
No.
We sent some DNA, even the original DNAs, I think I sent them to ANSES. And I kept the soil samples for many years, but afterwards I said, okay, so let's discard that because I cannot keep all these things in the lab. Normally I never throw away samples, but in this case I could not keep them on ages.
Maybe. Maybe next time I will just go around the island with David and just sample, take a piece once and to, just to see very small scale.
Non, mais ça va marcher. Alors les sols, on a testé, c'était le premier contrôle qu'on a fait. On a testé le sol direct et après culture. Ce qu'on avait vu, c'est que pour les amis, il fallait absolument enrichir en culture.
Il n'y a que des amis.
Théoriquement, oui, il n'y en a pas, il n'y a pas grand-chose. Il faudrait
Il n'y a que des amis dans les échantillons, les amis, ils ont tué.
Non, mais c'est parce que là, c'est enrichi. Mais ça, c'est les échantillons. Il faut retourner bien. Je ne sais même pas où c'est, mais toi, probablement.
Je les ai regardés, je crois, pour chercher l'histographie.
Ah, d'accord.
Ah oui, d'accord.
Mais tu n'as pas regardé dans ta
Non,
donc, du coup, mais si tu as un chiffre, ça ne va pas marcher. Parce que c'est vrai que.
You can check that.
Great.
Direct soil.
Okay. C'est tout. Merci beaucoup.
Merci. Thank you, Thomas.
So it was very, very long, perhaps, no? Yeah.
Do you want to take a picture? We will speak.
Okay.